Hi everyone, we're ready to wrap up chapter 4. So we will be covering section 4.5 and 4.6 in this lecture. So let me begin by sharing my screen and going to the PowerPoint. We're going to be discussing nucleic acids and the role they play in helping a cell produce proteins. We'll also talk about replication of the DNA that we've talked a bit about already last week when we talked about the different stages of the cell cycle in lab. So you might remember from chapter 2 on the chemistry chapter where they talked a little bit about the organic compounds. Those were the carbohydrates, the lipids, proteins, and nucleic acids. So we're talking here, of course, about the nucleic acid family of organic compounds. Specifically, we're going to be talking about DNA and RNA. But whether we're talking about DNA or RNA, both of these types of nucleic acids rely on building block molecules, which when put together anabolically make bigger, more complex molecules. And of course, here you see that building block molecule of RNA and DNA called a nucleotide, composed of 3 major constituents: a phosphate group covalently bonded to a sugar, that's what this S stands for, and you might notice it has the shape of the Pentagon building in Washington. This is called a pentose sugar, a 5-sided sugar. And then that in turn is covalently bonded to a nitrogenous base, indicated by the letter B here. So that, in a nutshell, is sort of the bare bones skeleton of a nucleic acid nucleotide. Now, these nucleotides can vary a little between DNA and RNA. We'll talk more about those in just a few moments. You might actually have already learned about that, I would hope, even in your high school biology class. When we start to link these nucleotides together, as we've done on this diagram, you can see that there are, what, 2, 4, 6, 8, 9 nucleotides that have been anabolically put together to form what's called a polynucleotide. Poly simply means many nucleotides, right? And we can see that this polynucleotide has what is referred to as a backbone to it. And it's formed as a result of these sugar phosphate groups, which of course I've circled here in red. So the backbone of a nucleic acid is made up of sugar and phosphate groups. When we talk about DNA, which I think, again, you are all somewhat familiar with, we often visualize it in the form of a double helix, right? I think you all have seen that. Well, let's take that double helix and let's unwind it and lay it flat against a surface, sort of like a ladder with rungs on it, okay? With the sides of the ladder being the sugar phosphate backbones, and the rungs of our double helix laid flat formed as a result of what are called hydrogen bonds between the bases. So here is DNA, obviously. You see the sugar phosphate backbones here on the left and on the right. You may notice that these are turned upside down compared to the left-hand polynucleotide. And that's because DNA is said to be anti-parallel. I won't get into the bonding reasons for that, but it has to do with the bonds forming between the sugar and the phosphate groups between adjacent nucleotides. So for the time being, let's focus our attention on the fact that we have these hydrogen bonds between the bases, between the nitrogenous bases. You might remember this from chapter 2. Hydrogen bonds are weak individually, but collectively they're quite strong. And this is a great example of how the double helix is held together thanks to these weak but collectively strong hydrogen bonds in the very, very center of the double helix, which again we're looking at here as having been unraveled and laid flat against the page, if you will, in the form of like a ladder. So I know this has to be DNA. This cannot be RNA. RNA is single-stranded, a single polynucleotide. In DNA, you have two polynucleotides held together in the middle by hydrogen bonds. Here's another representation of DNA. We see here at the top what we just saw in the previous slide, where we have the polynucleotides here on the right and left. Here's our bases in the very center. Now we're actually starting to give names to these bases. You might again remember this from high school or from one of the earlier sections of the textbook. G stands for the nitrogenous base guanine, and it's hydrogen bonded to another base on the opposite side. This is called cytosine, C for short. T stands for thymine, A stands for adenine. And so you always have this complementary base pairing in the center of the double helix. If there's a T on one side, there has to be an A on the opposite. These again are held together by hydrogen bonds. If there's a C on one side, on the opposite polynucleotide of the DNA must be guanine. This is referred to as Chargas rules. So you should memorize this, T to A and C to G bonding. When we take that double helix and begin to coil it around special types of proteins called histones, we see that it forms a material called chromatin. We talked about chromatin quite recently with respect to the form that the DNA takes on in the nucleus of a cell, right? And we said that when that cell goes to divide during the S phase of interphase, what happens to the DNA? Well, it replicates, right? And so that's what's happened here. Here's a sketch at the very bottom of this slide of a chromosome, a replicated chromosome that involves the loose strands of chromatin becoming very highly condensed and compressed to form a chromosome. And we know this is a replicated chromosome because what do we have here? We have two sister chromatids, right, held together at the centromere. So a chromosome is nothing more than a highly compressed, condensed form of chromatin. It's made up of DNA and associated proteins. Okay, I'd like to show you this short little video that goes over DNA. It'll discuss the complementary base pairing rules and so forth. So let's watch this.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities.
You don't need to know that.
The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves.
Don't need to know
These
that either.
grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves.
Okay, we're going to stop there. You don't need to worry about that stuff. Okay, let's go on to the next slide. We want to talk about the process of replication of DNA. Now that we have a good sense of what DNA is made up of, in terms of the complementary base pairing there in the very core or center of the molecule, right? G opposite C, T opposite A, with our sugar phosphate backbones on the sides. Let's go back and think about our discussion that we had recently in lab, where we talked about how in the S phase of interphase, we have replication of the DNA, right? We talked about that. This diagram is, of course, showing the cell cycle with interphase shown here. And of course, here's the 4 stages of mitosis, followed by cytokinesis, where we begin with 1 parent cell and end up with 2 daughter cells. So let's take a little closer look at the S phase of interphase, which is when the DNA replicates. So here we have the original parent DNA shown with the C opposite G and A opposite T rules. And what's happening here, as this DNA becomes replicated, the cell utilizes a special enzyme that I've noted here in red called Rna Polymerase. And what that DNA polymerase basically does is it comes into the molecule at a particular site, and it begins to break the hydrogen bonds here between the nitrogenous bases. sort of like unzippering a zipper, okay? So if we assume with my jacket here, we have the complementary base pairing here forming the teeth of the zipper. Here comes DNA polymerase, my hand, and I'm going to break those hydrogen bonds, and I'm going to liberate the two sides of the parent strand. And that's what we have here in dark blue. Here's one polynucleotide with its sugar phosphate backbone, shown here as the dark blue ribbon. Here's the other polynucleotide, shown by the dark blue. Here's, of course, our nitrogenous bases in the very center, but we have begun to separate them, right? We've unzipped the zipper. That hasn't happened up here yet, but it will as we proceed from bottom to top. So we have now these exposed bases of the original parent DNA, and what happens during replication is the cell synthesizes new daughter DNA nucleotides. Here we see two coming in, and they hydrogen bond to the exposed bases of the parent. This hasn't happened on this side yet, but you can see it happening here. So here's a nucleotide carrying guanine. It's going to hydrogen bond to the cytosine. The sugar phosphate here is going to connect to the light blue strand here. And here comes another nucleotide coming in. Doesn't say the letter here, but you should know what letter this is going to be, what base. If it's G here, it's going to be cytosine, right? And these daughter nucleotides that the cell is synthesizing, they come in and they bond to the complementary exposed bases of the original parent DNA. And this DNA polymerase continues to unzip the parent DNA until it comes to the end of the molecule. And what's the end result? Well, the end result is going to be the formation of two double helices of DNA, right? If you look at either of those, half is parent, i.e. dark blue ribbon, right? And half is going to be daughter DNA, shown here in light blue. But when you examine the DNA molecule here closely, you find the same base pairing. That's why we say that there's been replication of the DNA. And you know that down the road, once we get past mitosis, we end up with the two daughter cells here. We've said that they are genetically identical because these two double helices will be eventually divvied to the two cells as that replicated chromosome separates during anaphase. So hopefully you're starting to connect the pieces of the puzzle here. So that's DNA replication, an important process that occurs in the nucleus of a cell as it goes to divide. This occurs again during the S phase of interphase. Let's watch another short video here about DNA, and this will review the replication process.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated, and the DNA replication machinery is assembled at the replication fork.
Remember, what is separating these two strands? It's the enzyme Rna Polymerase.
The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the Atgc Rule, also known as Chargas Rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
Okay, a really nice summary of replication. Really like that.
DNA.
Okay, we're going to shift gears now and talk about the role that DNA and RNA play together to help cells construct proteins. Remember, proteins are a very important group of organic molecules. We know that some proteins act as enzymes, others act as transport vehicles through which the materials can pass through cell membranes. Others are structural. They help provide strength to bone and other connective tissues. So your cells construct an enormous diversity of proteins, and they do that with the help of these two nucleic acids, DNA and RNA. So let's take a moment and compare and contrast the two molecules. We already have talked a little bit about this, but this is a nice review. The first thing to note is that if you look at the nucleotide of DNA and compare it to a nucleotide of RNA, it's still got the three major constituents, right? The phosphate group, the sugar, and the nitrogenous base. The phosphate group between the two types of nucleic acids is exactly the same. The sugar, however, is different. And as this table sort of implies, in DNA, the sugar has a special name called deoxyribose sugar, while in RNA, an RNA nucleotide would have a ribose sugar. And the reason that they're different is because off of, I think it is the second carbon of the sugar, there is not an oxygen. And that's where the term deoxy comes in the DNA sugar. In an RNA sugar of the nucleotide, it has an oxygen coming off carbon number 2. So that's the only difference. Otherwise, the pentose sugar is the same. And you don't need to worry about the chemistry of it. Just wanted to denote that there is a difference between the sugars of the two types of nucleic acids. Here is where the other extremely important difference comes into play, and you need to know this. In DNA, we talked about the 4 possible nitrogenous bases: cytosine, guanine, adenine, and thymine. And we talked about, as the video did, that there's this complementary base pairing in the very center of the double helix. If you have a C here, you got to have a G opposite it. If you have an adenine, opposite it will be thymine. Commit that to memory. It's called complementary base pairing, or Chargas rules, named after Erwin Chargas, who was a biologist who discovered this back in the 1960s. In RNA, you have 3 of those same sugars. And you see those listed: cytosine, guanine, and adenine. But there's no thymine. In place of thymine, we have uracil. There's also not a double helix here, okay? So we're going to talk more about this in just a moment, but just know that there is a difference in the types of nitrogenous bases between RNA and DNA, and that in DNA, you have the double helix, in the center of which are the complementary base pairing. The other thing that this slide notes is the fact that you have 3 different kinds of RNA. And I listed those in this blue box: messenger, transfer, and ribosomal. Sometimes we just give them the abbreviation mRNA, tRNA, and rRNA for the different kinds. So just remember, M is messenger, T transfer, R ribosomal. Let me just put ribosomal RNA to bed right off the bat, because we're just going to make one comment about it. And that is, when you look at a ribosome, which you know is an organelle found in eukaryotic cells, what occurs at the ribosome? You should know this. What important process takes place? If you answered protein synthesis, or the construction of proteins, the assembly of proteins, you're right. So that ribosome, whether it's freely scattered in the cytoplasm or associated with the rough endoplasmic reticulum, that's why it's called rough, because it has the ribosomes on its surface. That ribosome is composed to a large extent of rRNA. Okay, that's all you got to remember. Ribosomes are made up of rRNA, and it's where proteins are going to get assembled or manufactured. It's the site of protein assembly. This is going to review what we just talked a little bit about a moment ago. When you compare RNA to DNA, what are some of the differences? Well, we've already talked about how the sugar is different, right? Ribose instead of deoxyribose in RNA. Is the phosphate group different? No. Are the bases different? There is one base difference, isn't there? There's no thiamine, is there, in RNA? In place of it, we have uracil. The other thing that you note is that RNA is not double-stranded. It's single-stranded. So we're looking here at a polynucleotide of RNA, sugar phosphate backbone, okay? But we don't have any complementary base pairing rules here applying, do we? Because it's single-stranded. And the reason that I know this is an RNA polynucleotide would be if I examined the sugar closely and looked at the number of oxygens and hydrogens coming off of these carbons shown at the corners of the pentose sugar, I would be able to tell that coming off carbon number 2, I'd have an oxygen. So that would tell me it's a ribose sugar and I have an RNA molecule. We can't see that here, but if we looked into the chemistry, we could deduce that. What is extremely obvious here, though, that tells me this is an RNA polynucleotide is the presence of the uracil, okay? Because only RNA has it. And I pointed out a couple of them here. So this is an RNA molecule, single-stranded. Okay, important to note those differences. I'm going to show you a very short video here on a process that occurs at the very beginning of protein synthesis. It's called transcription. And I'm going to walk you through some of the steps, but I'd like you to watch this video. You may need to watch it a couple times, but let's just sit back, watch the first minute and a half about of this video, which will take you through the first step that cells undergo as they begin to construct proteins. Remember, both DNA and RNA are going to play an important role in that process. In fact, DNA is going to act as a template for the formation of messenger RNA. That's basically what happens during transcription. So let's watch this YouTube video.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location.
Okay, so good point here to understand, and that is the process of transcription, the first step of protein synthesis, begins at a particular site on the DNA called the promoter region and will end at a particular site on the DNA called the terminator region. So we're going to be looking at what happens between the promoter and the terminator region of the DNA.
Distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind.
So now we're going to talk about a close cousin of DNA polymerase called RNA polymerase. Don't get them mixed up. DNA polymerase was instrumental in allowing for DNA replication. RNA polymerase is going to allow for transcription, the first step of protein synthesis. Do not get them mixed up. So this big pink guy is denoting an enzyme, and I know it's an enzyme because it ends in ASE, right? Any word that ends in ASE is an enzyme. And this enzyme is going to start to interact with the DNA at the promoter region, and then it's going to move along it. Let's watch what happens.
This is where the majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay, so what we're left with is this long polynucleotide called messenger RNA that has been transcribed from the DNA. And so you can go back and watch this short little video as many times as you want. Let's talk about what's happening. So I'm going to the next slide here. Here we have the double helix of DNA found in the nucleus of the cell. So this is the nuclear envelope here. Here's the cytoplasm as denoted on the slide. And then we have a cell membrane way out here somewhere. The first thing that happens, as the video mentioned, is RNA polymerase will associate with a certain region of the DNA called the promoter region. It's not labeled on here, but let's pretend it's down here. And then it will continue to interact with the DNA until it comes to a terminator region. What happens between promoter and terminator? Well, the video talked about how the RNA polymerase does two major things. It breaks the hydrogen bonds here between the C and G and T and A. That's happened here. So here we can see half the DNA on the left. Here's the other half that I'm noting on my cursor here on the right. In addition to separating the double helix, the RNA polymerase will also begin to bring in RNA nucleotides one at a time. So if we have a G here of DNA, we will be bringing in a RNA nucleotide that's carrying cytosine. If we have an exposed a T rather, then we'll have a RNA nucleotide that carries the base A. So we're following the complementary base pairing rules until we get to this scenario. If we have an A exposed here on the template strand of the DNA, we don't have a T brought in because there's no thymine in RNA. Instead, the cell would bring in a nucleotide carrying uracil. Now, again, these are brought in individually, but then the sugar phosphate groups fuse, so we end up making a big, long polynucleotide. There is temporary hydrogen bonding between the RNA base and the DNA base, but eventually that hydrogen bond breaks and that big RNA polynucleotide will dissociate from the DNA and the DNA will coil back together again. No harm done. What has occurred is simply that the DNA strand here, we call it the template strand, is being used to form the messenger RNA. This other strand here on the left plays no role whatsoever in helping to construct the messenger RNA. It's just this side. And we got to remember that if we have a C here in DNA, we're going to have a G in the RNA. If we have an A in the DNA, we have a U in the RNA. If we have a T in the DNA, we'll have an A in the RNA. If we have a G in DNA, we'll have a C opposite it in the RNA. So this sequence of bases of the RNA, the messenger RNA, that's determined, isn't it, by the base sequences of the DNA. So the DNA is this master molecule that's giving instructions for the construction of this big, long polynucleotide called messenger RNA. And this process, again, begins at a particular region called the promoter and will end at a particular region called the terminator. And the RNA polymerase slides off to the side, the DNA coils back together, and here we are left with this long strand of messenger RNA, which is going to leave the nucleus. That's what's going on here. We see this messenger RNA that's just been transcribed from the DNA. It dissociates from the DNA coils back together, and this RNA leaves the nucleus and goes out into the cytoplasm. So important to note, this process occurs in the nucleus. And again, we've already talked about the base pairing between the DNA and the messenger RNA. And remember, no thiamine in messenger RNA or in any kind of RNA. In place of thiamine, you got to put a uracil. Okay. Now, let's take a closer look at the messenger RNA polynucleotide that we just transcribed from the DNA. Remember, it's left the nuclear envelope, has gone out into the cytoplasm, and we'll pick up in just a moment where we left off, what happens after that. If we look at that messenger RNA, what we'll notice is that every linear sequence of 3 bases has a special name. It's called a codon. Codon. So we're looking at 7 codons, or 21 individual nitrogenous bases in this RNA polynucleotide. This blue structure here is representing the sugar phosphate backbone, right? We don't see those here, but we assume and need to understand you have the sugar phosphate backbone holding that polynucleotide together with the base bonded to the sugar, right? And then we have the phosphate and sugars making up again that backbone. So we're just focusing here on the bases, the nitrogenous bases. And each of those codons that you see here, the A's, the U's, the G's, and the C's, whose sequence was determined by the DNA, each of those unique combinations codes for a particular amino acid. There are 20 or 20 plus amino acids, and that's the names we see here, these funny looking names, methionine, glycine, serine, and so on. Those are names of individual building block molecules of proteins. You remember that from chapter 2, I hope. Amino acids are building blocks of proteins. So these codons will represent particular amino acids, which were denoted here by the different colored, sort of like beads of a chain almost, isn't it? So keep that in mind. Each codon, each linear sequence of 3 bases, RNA bases, represents one of 20 or 22 different kinds of amino acids. In fact, here we're looking at a table that you can find in your book. There on page 143. What we have here are all the possible codons that could be created in messenger RNA. There are 64 possible ways you could arrange 4 different bases in triplets. 4 to the 3rd power is how it's calculated. 4 to the 3rd power is 64. And so you'll see that many of these codons can code for the same amino acid or represent the same amino acid. In other words, these two, UUU or UUC, represent phenylalanine, that amino acid. Here are 4 codons that represent the amino acid leucine, and so on and so forth. You do not need to memorize this table, but I think you have to understand how it's developed, what it means. I will point out just 4 here that the textbook has circled, and I'll go over this again later, but I just wanted to point out, you might be wondering, what's the circled ones? Well, this one here, AUG, the abbreviation for the month of August, is sometimes called the start codon. It represents the amino acid methionine, or MET for short. These other 3, UAA, UAG, and UGA, notice they say stop. They do not code for any of the 20 plus amino acids, but rather will play an instrumental role in stopping protein assembly. Okay, so they're just the red light that comes up. This is the green light that kind of gets the process rolling. So we're going to talk about that process next. It's called translation. So we've just talked about how messenger RNA is transcribed from the DNA in the nucleus and how that messenger RNA has left the nucleus, gone out into the cytoplasm. There it will interact with the ribosome, which you know is made up primarily of rRNA. Okay? So the ribosome, remember, is where the protein is going to be assembled. Protein synthesis occurs here. And we will also talk about the third player, the third type of RNA called transfer RNA. And as the slide indicates, this is going to play an important role in helping to shuttle individual amino acids to the ribosome, which then help to make the protein. So here we are looking at a ribosome shown here in brown. Okay, there's actually a large subunit and there's a small subunit, but let's just call it ribosome. Here's the messenger RNA that we just transcribed in the nucleus, and it has associated with the ribosome, connected to it, actually. And these kind of funny blue-looking structures are two transfer RNA molecules. And as the name sort of implies, these transfer in individual amino acids. Now, in this particular view, we have a transfer RNA shown here by the green arrow that is connected to 5 amino acids. We'll talk about how those got there in just a minute. This next transfer RNA has come in and it's carrying an amino acid, number 6, that will be added to the growing polypeptide chain. But what I want you to center your focus on here, your attention to, is the codons here in the messenger RNA. We already talked about what those are, the linear sequence of 3 bases. Notice this codon number 5, ACG. This part of the transfer RNA is called the anticodon. It carries the 3 bases that will complement the bases here of the messenger RNA codon. And remember, if you have an A, opposite it will be in RNA a U. If you have a C, opposite it will be a G. If you have a G, opposite will be a C. So the anticodon of this transfer RNA carrying number 5 is complementing the base sequence here of the messenger RNA. The same thing has happened already here in the next codon, whose sequence is GCA. That transfer RNA that's brought number 6 to be added has to have the proper anticodon sequence to complement the codon here of the 6th codon listed. So GCA codon must have anticodon CGU in that sequence, and this transfer RNA has that, and therefore can combine with the messenger RNA at the ribosome. And that has already this has already happened in this next diagram. Now notice what's happened though is we have brought that 6th transfer RNA in, whose anticodon matches the codon sequence in number 6 codon, and is carrying amino acid number 6 to be added. Can we determine the amino acid? We call it number 6, but can we find out what the name of that is? And the answer is yes. We can determine which amino acid number 6 is by looking at the codon sequence here of the messenger RNA, which reads GCA, right? GCA. So if we go to that table, to GCA, it tells us that represents the amino acid alanine. So there's the answer to our question. Number 6 is alanine. Now you may have also noticed that we have formed a bond here between alanine and the preceding amino acid called a peptide bond. Gotta connect these two guys together. And then we've got to disconnect this transfer RNA and the one that it was carrying, number 5. Let's get that guy out of there. So we break that. This guy is going to leave minus its amino acid and pick up another one. And if it's anticodon sequence, UGC complements a codon sequence down the line here, which would be ACG. It'll simply shuttle another amino acid into place. So this is just shuttling or transferring amino acids, thus the name transfer RNA. But it now leaves, we break the hydrogen bonds here between the bases of the codon and the anticodon bases. This guy leaves. Here comes another amino acid carried by another transfer RNA molecule. But before it can come in and lock in place, we have to move the ribosome over one codon sequence. So the ribosome actually shifts, it moves along the messenger RNA. And this next transfer RNA is going to come in. How can we determine the identity of this amino acid? Well, I hope you know the answer. We look at the codon sequence here of the messenger RNA, which reads GGC, okay? And we know the anticodon must be CCG, or this guy wouldn't come in. And we look then on that table for GGC. There it is, and it's telling us it represents the amino acid glycine. So this is glycine. Number 7 is glycine. So in essence, what's taking place here in translation is the ribosome shifts along the messenger RNA, and at that site where the ribosome and the messenger RNA interact with one another, individual transfer RNAs bring in individual amino acids. Their anticodon sequence must match the exposed codon sequences of the messenger RNA. We then link these amino acids together. So as she says, this is growing. We are growing this polypeptide chain one at a time as the ribosome moves along the messenger RNA, and individual amino acids are shuttled in by individual transfer RNA molecules. So let's watch this next video.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA.
So this is simply describing the initial steps of translation. Okay, we're getting the messenger RNA associated with the ribosome. And you can see the big subunit coming in here. Here's the small subunit. And then this is our first transfer RNA coming in.
The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown.
Now, I want you to look very closely at this first codon sequence, AUG. What did we say about 5 minutes ago with respect to the table that had all those codon sequences? AUG, the abbreviation for the month of August, was the start codon, and also represented the amino acid methionine, MET for short. So there's the start codon that's starting the translation process. This blue sphere is representing the amino acid methionine. Notice the anticodon sequence of this transfer RNA must complement the sequence of the codon, and it does. Okay, let's start to create the polypeptide.
The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site.
You do not need to know P and A site, but I think it's helpful to see kind of how this process occurs. So it's worth noting anyway, but I'm not going to ask you P site, A site. but just make note of the role they play.
The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site.
Now, this particular transfer RNA that's brought in the green sphere, which again is another amino acid, how could we look that up in the table in our book? We'd have to look up CCG. And if we did that, we'd find that this amino acid is the amino acid proline, PRO. So we have connected these two together. We've created a peptide bond. This transfer RNA is now going to leave minus its amino acid, right? And go pick up another methionine and bring it into the protein if it has that particular match set down the road, AUG. It'll bring in another methionine.
The ribosome now advances a distance of 1 codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon.
So this is one of those 3 stop codons I mentioned a few moments ago, which tells the process we are done translating. We're done making the protein, because no transfer RNA is going to recognize that.
The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Here's our polypeptide. Now, this is only showing maybe about half a dozen amino acids in length. The reality is that most polypeptides are hundreds, if not thousands, of amino acids in length. In order to create a functional protein, it's got to be quite long, not just 6 amino acids long, but hundreds, thousands, sometimes even tens of thousands of amino acids comprise a functional protein. Now, what can often happen too, which is really interesting, and they're showing the dissociation of the ribosomal subunits here, which pretty much, again, ends translation. You can have multiple ribosomes going one after the other down that same messenger RNA strand. And the result is the cell can construct a whole bunch of the same protein. Remember, the ribosome is the assembly site of the polypeptide or protein. So you can have multiple ribosomes, moving along that same messenger RNA and creating a lot of the same protein.
Translation is initiated.
Okay. So again, this simply reviews the presence of the 3 stop codons, UAA, UAG, and UGA. So you might want to know those. And here's a review of that process of elongation, it's called. It's a subset of translation. It talks about how the polypeptide is elongated or being constructed. It's, again, the movement of the ribosome along the messenger RNA and the bringing in of individual amino acids by individual transfer RNAs. And then here again is a more broad review of both steps of protein synthesis, transcription, followed by translation. So you should be able to kind of look at this and glean a lot of good review from the moment the DNA is transcribed to the moment that a cell constructs the polypeptide. Remember, everything starts at the DNA. The DNA is what provides the information for the ultimate production of the protein, because it's the sequence of the DNA bases that determines the sequence of the RNA bases, the messenger RNA bases, which in turn determines the sequence of the amino acids. So, very good summary slide. We want to wrap up chapter 4 and talk a little bit about what happens if there are changes that occur to the DNA. You've all heard of mutations before, haven't you? And we have a negative sense of what effects they have on a cell, and they certainly can have very bad effects to the cell. Not all mutations are bad necessarily, but mutations, let's just think of them for right now as changes that occur to the DNA. Now here we're looking at a very small segment of the template strand of DNA. So to set the stage, we're talking about transcription has already occurred. We've separated the two DNA strands. This is the template strand that's going to be read as the messenger RNA is constructed. Okay, so we'd have another strand of DNA over here that does not play a role at all in transcription. This is the template strand that is being transcribed into messenger RNA, and we're just looking at one codon sequence here of the DNA. Here's the corresponding complementary codon of the messenger RNA that would be transcribed. So if we have CTT on DNA, opposite it, we'd expect to see GAA as the messenger RNA codon created following transcription. If you look up GAA in the table on page 143, you'll find that it represents the amino acid glutamic acid, or GLU. Well, what happens if we change the DNA? What if we alter it? Okay, this next video is going to talk a little bit about that, and then we'll talk more about the process at the end. So let's watch this. There are different kinds of mutations.
A mutation occurs by base substitution when an incorrect base is incorporated into DNA. Some base substitutions occur because purines and pyrimidines exist in two structural forms. The most common form results in base pairing between adenine and thymine and between guanine and cytosine. However, the hydrogen atoms can move to form a base with altered hydrogen bonding properties, creating a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can bond to cytosine. A tautomeric shift in thymine allows it to bond to guanine. This error in DNA replication is passed on to the cell's progeny. The change in a single nucleotide in the DNA results in a change in the corresponding nucleotide in messenger RNA. The change in the codon can result in a different amino acid being incorporated into the protein.
So we've said up to this point that we have always had the A opposite T and C opposite G pairing, right?
Mutation occurs by
So you go back to this last part of the
base substitution
video.
When an incorrect base.
Okay, I'm going to stop it here. You can get these weird combinations on rare occasion as a result of a mutation, where instead of having AT, we have a GC a GC combination. We introduce a pair, so that's why it's called base pair substitutions. We introduce a different pair of nucleotides, and the effect is to form a totally different codon, which in turn will result in a totally different amino acid being brought in down the road. So let me go to this next slide. I think it'll do a better job kind of describing the process. Okay. So here we have a change to the middle base. Go back 2 slides and look at that preceding slide. We're substituting a different base here. Instead of having a T, the mutagen, the mutating agent, which could be a variety of things. It could be a chemical. Chemicals can act as mutagens, inducing mutations. You've heard of UV light increasing the risk of skin cancer. UV is a mutagen. So anything that can cause a mutation or a change in the DNA is a mutagen. So we're going to represent that mutagen by a lightning bolt. We're going to change that original thymine now to an adenine. That can alter the amino acid that would be coded for or represented by that codon. So instead of having, the previous sequence, which I forget what it was now, let me go back here. Instead of having CTT and having GAA being transcribed, now we have CAT, CAT, which will result in a totally different amino acid being represented. So if you look up CTT, you'll see glutamic acid. If you look up CAT, you're going to see it codes for a different amino acid altogether, valine instead of glutamic acid. And you might say, well, okay, we're bringing in one different amino acid. What difference could that possibly make? Well, it depends. Lots of things can influence this. But what could potentially happen with a significant difference between having a normal versus a mutation, a normal sequence versus a mutation sequence, could be the difference between having a normal biconcave red blood cell, like we see here, or having what we call sickle cell trait. So somebody that has sickle cell disease, or we used to call it sickle cell anemia, that's caused by a single point mutation, a base pair substitution that goes into the construction of the cell in the bone marrow. So without the mutation, the bone marrow creates nice, healthy, biconcave red blood cells that transport oxygen and CO2, no problem. But if you have the wrong base put in, one base creates a different amino acid being brought into the polypeptide, and that creates the weird sickle cell shape of the red cell. This can clog up capillaries. It can impede gas exchange between blood and tissues. It can cause pain. That is a pretty significant impact, I would argue, isn't it? Well, luckily for us, even though mutations are very rare, but they do take place, okay? We have the ability, our cells have the ability to detect those mutations, those mismatches, the A opposite C or the G opposite A, which shouldn't happen but can. When our cells undergo mutations, we have special proofreading capabilities, they're called proofreading enzymes, that can scour, for example, the newly synthesized or newly replicated DNA and find the mismatches and correct them. So if there's a G and a T, let's snip out the T and put a C in, and then we'll have no problem connecting those two. The DNA will be basically repaired. Yeah, that's pretty cool. So to summarize, while mutations are exceedingly rare but could be life-threatening to a cell or an organism, most of the time we have these special repair enzymes that can recognize and remove mismatches and put in the proper base so that no harm is done in the long run in terms of the protein that's going to be produced. So we've got our lecture exam coming up on Thursday. Just want to remind you of that. 80 points over chapters 1, 3, and 4. Nothing on the chemistry chapter. And your choice here of a couple different essays, but definitely multiple choice, matching, possibly some true false, maybe definition, short answer. a potpourri of types of questions, but you will have your choice of a couple essays. I think I'm asking for like 2 out of 3 essays on the test. So if you haven't started studying for this, you should have by now. Do not wait until Wednesday night. Do not do that. It will not turn out well. You should be studying, weekly, if not daily, really, on this, keeping up with the material. Okay, this wraps up chapter 4.